Okay, everyone, welcome back to our last session of the day. We're going to be talking about aquatic invasive plants for this final session. So looking forward to that. Thank you to our speakers for joining us this afternoon. Em's going to be taking us through again, but before I pass it to her, I just want to send one final reminder that we will have all of the sessions that are recorded available on our YouTube channel to watch at a later date. So if you've missed anything along the way this week, definitely check it out there and you'll be able to hopefully find what you're looking for. With that, Emily, I'll pass it over to you to lead us through to the end of the day.
Okay, thanks so much, Mackenzie. Hope you all had a good break. In case you're just joining us now, just want to let you know to please put any questions you have for our speakers in the Q&A box. I will then read those out loud and we'll try to answer as many of those as you can. But without further ado, I would like to introduce our next speaker, Michael Mctavish. He's coming to us from the University of Toronto to speak to us about Phragmites.
Okay, great. Thanks very much. We're in business here.
Yep, looks good. Thanks.
Okay, perfect. Well, good afternoon, everyone. Happy to be joining you here. I'm going to be speaking on behalf of myself and colleagues here as well, providing a bit of an overview of these first several years of the biological control program for invasive introduced Phragmites here in Ontario. So our weed in question is, of course, introduced Phragmites. I'll be referring to it as such. Specifically, we're referring to the invasive non-native subspecies, Australis australis, and this is, of course, considered one of the most invasive plants in North America. This is a tall perennial grass that forms these very dense monospecific stands that have various economic and infrastructural impacts, as well as ecological impacts, which include threatening a native subspecies of Phragmites as well. There are various control options that are available for Phragmites, but these can vary in efficacy and efficiency in different contexts. And so there is always a need for additional options to supplement the management tools that we already have. And the tool that we are offering here is biological control or biocontrol. Broadly, this refers to the use of live organisms to suppress an introduced pest. Some of the appeal for using biocontrol is that it is attempting to reunite these pests with the natural enemies that they have escaped from that would normally help keep their populations in check. Because these natural enemies are selected to be highly host specific, it can be very targeted and have low environmental off-target impact. Biocontrol can become self-sustaining as the organisms reproduce and track the host through space and time. And because of that self-sustaining nature, this can be very cost-effective, even at very large spatial and temporal scales. So the biocontrol program for Phragmites is something that's been in development for over 20 years now. The tool that we're offering here, this sort of additional tool in the toolbox, if you will, uses these two European moths with stem-boring larvae. So shown here as caterpillars, and as moths, we see Archanera neurica and Lanisa geminipuncta. The larvae are the life stage that do the actual damage. They mine shoots and will switch between multiple stems through their life cycle, causing stem wilt and mortality. And this has the net effect of reducing overall competitive growth and reproductive output of the Phragmites. We'll give here a general life cycle of the two species. They're slightly different, but similar enough. We're going to do them in one here. The larvae hatch in the spring and will feed in the shoots for about 4 to 6 weeks. And again, this is the period where it's doing the actual damage to the target. They pupate in the stems themselves for about 26 to 40 days, and then they emerge as short-living adults, flying and mating, and the females will lay approximately 100 to 150 eggs under the close-fitting leaf sheaths of the plant, which is where they will overwinter protected until the following spring. This program began in 1998 with explorations for suitable biocontrol insects. Over time, these were selected and then subjected to an extensive period of host range testing to ensure that they are specific to the target weed and that they're not going to be targeting anything else. This work culminated in petitions to release in Canada and the US in 2018. That US petition is still under evaluation as it's a more extensive or just longer process. And then this was approved in 2019 in Canada by the CFIA, allowing for the current Ontario release program that we've been involved with. The first years of this program have really been focused on developing these core methods because this is a new program. We like to organize these by thinking of this in terms of methods to rear the biocontrol agents, to release them onto the landscape, and then to monitor their eventual establishment and impacts. And all told, that has allowed us to put together this first operational protocol for biocontrol of invasive Phragmites. So I want to give a bit of a look here of what that protocol looks like that we've now developed. So again, focusing on these stages of rearing, release, and monitoring for these two agents, I've put these steps on this timeline layout here, color-coded by these three different steps, and we're going to go through them in order here. Starting with rearing. So you need biocontrol agents to actually do anything in the first place. Rearing typically begins in the spring, around May, although, as we'll see, you can do this at different times of the year as well. Usually we're rearing from eggs, so the top middle there. These will hatch out into larvae that, depending on the species, we can feed on cut Phragmites stems or an artificial insect diet, and then this can go on to pupae and then to moths. So we can stop this process at any life stage to release that life stage. So for example, we could stop at larvae or stop at pupae, or we could continue it through and then get more eggs from moths to continue this cycle. And because this can be done indoors in the lab under lab conditions, rearing can be near continuous. So by staggering different generations, we can stockpile up to 2 or 3 generations per year and then store that for a larger release at the end. If we look to sort of mid-July, we're going to talk more about release in a moment, but what we really look to are what we call nurse sites. So these are locations where populations become established and are reproducing, and these become important sources of additional insects for further rearing and redistribution. So for example, as seen here, we can cut some of that Phragmites stem biomass and then harvest that to relocate or pull the eggs off of them for additional rearing or distribution. Once we have the eggs, they then need to be stored for the remainder of the year through till the following spring or until whenever they're going to be used for continuous rearing efforts. We found from experimentation that we get the best hatch rate of those eggs if we store them under naturally fluctuating outdoor conditions. So that's what we're doing now. Generally, we'll put the eggs into a semi-sheltered but outdoor environment, which is going to give them the best hatch rate possible. Once we have the insects, we need ways to release them onto the landscape. These releases typically occur in the spring or early summer with specific timing depending on what method we're using. Both of these agents can be released at all life stages, so as eggs, larvae, pupae, or moths. These require different timings, but also different methods. And generally, these will involve some trade-offs between efficacy and efficiency that we'll get into. The earliest releases would happen with eggs. Our current method is to introduce eggs into basically these plastic cups that have a screen mesh bottom. That mesh helps keep predators out, but the larvae, as they hatch, can climb out through that mesh and make their way out into the patch. This is a really efficient, quick technique for releasing large numbers of insects, and they are offered some amount of protection from weather and predators in that vulnerable egg stage. Later on in May, in towards June, we can also release larvae. The best method that we've found to do this is to allow eggs to hatch out into larvae and then to inoculate those larvae by hand into cut Phragmites stems. This gives them something to feed on and sort of a sheltered environment for a period of time. We can then collect a bunch of these larvae inoculated stems into blocks of foam to keep them moist and then put those out into the field. So they'll feed in those stems for a period of time, then chew their way out and switch to a new stem in the field. This requires quite a bit more effort. But is currently one of our most effective methods for getting reliable damage post-release. We can also release as pupae or as moths. So you can see here, these are containers filled with vermiculite that we use to release pupae and moths, and these might be out in the open or maybe in a caged environment to keep them contained. They are some of the most logistically demanding release methods, so it requires that extra rearing to get them to these late life stages, and you have to get the timing right, but they are also a viable option and something that we've used successfully so far. Once insects are on the landscape, we then require monitoring. This monitoring is important to compare the effectiveness of different techniques that we're trying to develop and to assess longer-term insect establishment on the landscape and ultimately impact on the target weed. There are two main monitoring time frames that we look at. So summer, we've learned, is kind of the optimal time to look for larval damage before the vegetation gets too tall. And then fall is the easiest time to come back to take measurements, looking at individual Phragmites, Ramites, and overall stand characteristics, so things like population density of the weed. So if we're looking in mid-June, sort of summer timing at larval damage, because they feed and pupate internally, we don't generally directly observe the biocontrol agents. More often, we are looking for indirect indicators of activity. So some of the symptoms that we would be looking for would include these larval boreholes, so where they chew their way in and out of the stems, the wilt of the stem tip that follows that feeding damage. There's this secondary branching that can occur as the plant tries to compensate for damage to this growing tip. They'll put out these secondary branches below that damage, and also pupal windows. So that's where the adults would actually emerge post-pupation from the stems to fly. If we're going into a site where we just released this year, we know exactly where we put them and they don't go too far that first year. So we can do a very focused monitoring, looking at the amount of damaged stems around a known release point. If we're looking at a site in the second year onwards, they've had the chance to potentially disperse as adult moths, which is what we want them to do, but we now don't know exactly where they're going to be. So we've developed a sort of more of a rapid screening protocol that looks at these 4 meter by 1 meter intervals that we will run using rope through and around Phragmites patches, and in each of those intervals, we can quickly quantify the presence or absence of feeding damage and quantify the number of damaged stems per meter of transect. And then, as mentioned, you can go back later in the fall. The insects have long since done their damage at this point, but we can now quantify these ramete and stand level responses in the fall when it's just easier to do that because there's less vegetation around. So, how are things going at this stage in terms of overall release progress? These first few years of ongoing trials and experimentation have led us to this operational protocol that we now have for Phragmites biocontrol. Again, looking at these stages of rearing, release, and monitoring. And here are all those steps put back onto that timeline. And it's worth noting here that there is a positive feedback cycle that we're trying to set up here, whereby the more releases that we do, the more nurse sites are able to put on the landscape. Those are then sources of eggs that go back into rearing and redistribution. And so again, we get this positive feedback cycle that we're looking to set up. Using this operational method, as of this winter, we now have approximately 17,600 biocontrol insects of both species released across 13 sites, and you can see them on the map there on a bit of a latitudinal gradient that we've tried to set up. We're trying to get good coverage at a landscape scale. If we look at that initial larval feeding damage, so this would be looking at sites where we've just released in that first year, immediately around that release point, and we're looking at the percent of stems around that point that have insect feeding damage on them. This is a really useful metric for assessing release success and giving us an indication of how that population is going to do longer term. And in general, what we see is about 2 to 39% of damaged stems can be found around that release point in that first year. It's worth noting that some of these sites we don't have data for because if you release as pupae or moths, there's not going to be any larval damage around that release in that first year. So looking towards year 2 and onwards, we're switching gears into that follow-up monitoring where we're quickly assessing insect damage in these 4 meter intervals. Again, if a site is relatively new, then we're not going to have follow-up monitoring data, so that's why some of those are NA. Of the sites that are 2 years or more in, out of 8 sites, we saw follow-up damage at 6 of them, with damage ranging from 6 to 100% of those intervals that we looked at containing at least one damaged stem. So seeing any amount of damage in this second year is extremely encouraging. It means that they've been able to complete at least one full life cycle and make it through an important overwintering potential choke point. And as you'll see here, we have a few sites that are sort of our superstar sites where we have very good damage coverage where they've spread substantially from these initial release points. So we're really excited to be seeing this at this point. And it's worth noting at this point too that across all those releases, we've seen different release methods all have success. There are sort of pros and cons to different ones, but all have managed to produce insect damage in that first year and second year post-release. So to quickly summarize here at the end and look at some next steps, in these initial years of the program from 2019 to present, working on Ontario, we have developed and now have in hand this operational biocontrol protocol for invasive Phragmites. Using that protocol, we've been able to release over 17,000 insects across a network of 13 sites, which we are looking to expand now. And we are able to detect insect damage immediately post-release and 1 year onwards post-release, which is having us be very, very positive and encouraged about seeing long-term population establishment and these transitioning to be these kinds of nurse sites that we can go back to harvest eggs from for further rearing and redistribution. So looking up with this protocol in hand, the focus now is really on scaling all of this up. So scaling up rearing and release of biocontrol agents across Ontario. This would be through a combination of the lab rearing that we described and also field harvesting of eggs from these nurse sites as they become nice and established. We're working with many partners on this throughout various aspects of this program. We just want to flag in particular, we're working closely with Ducks Unlimited Canada for key support of this scaling up process in the years going ahead. And as we do this, we're going to be looking at establishing an increasingly broad network of nurse sites across Southern Ontario. We're looking for as good coverage as we can, trying to set up, again, this positive feedback cycles of more releases, giving us more nurse sites, allowing more releases. And I should flag that as more of these sites become developed across the landscape and we get some of that dispersal that we are hoping and expecting to have, it'll be increasingly important to keep an eye open for potential biocontrol damage on your very own Phragmites as well. So as we noted when talking about monitoring, this is usually easiest to see in June. You're looking for that circular borehole or two in the stem. Usually what you see as you're walking through would be the wilted stem tip above that borehole is kind of what is flagged first. And very quickly as well, I'd like to flag that as a part of our broader Phragmites work, we also have a Phragmites identification tool that we're in the process of putting out. So this should be available very shortly. We are focusing on putting together a tool that identifies easy-to-use morphological traits to differentiate between native and introduced subspecies. The idea was to create a tool that is regionally specific to Southern Ontario from our survey work in this area to provide a very practical tool for land managers to use to complement genetic identification as well. So stay tuned for that shortly. Of course, this is a large-scale program that involves support from various partners and individuals, so we're very grateful for the support of all of them, and you can see them all here. And with that, I'd like to say thank you, and if you have any questions, we can address them now, and I also have contact info here if you want to get in touch. Thanks very much.
Thank you so much, Michael. We do have just a couple of minutes for questions. So our first question is, wondering if the moth/pupa show a preference to the non-native Phrag or if they equally predate all Phragmites.
Right. So looking at preferences of the moths between non-native and native Phragmites was an important focus of that host specificity testing. They are extremely specific to Phragmites australis overall, with a very, very strong preference for the introduced subspecies. Essentially, there are some morphological and physiological traits of the native Phrag that make it a very poor host, in particular the fact that the eggs get laid under that leaf sheath, which is shed by native Phragmites in the winter. So they don't lay eggs there very often, and if they ever do, those leaf sheaths fall off, and that small number of eggs are exposed for the winter and aren't able to survive that period.
Okay, thank you. I have time for maybe one more, but I think I'm going to combine two that are about Northern Ontario, if that's okay. Can your ID tool work in Northern Ontario, and when will Northern Ontario be a site for this research?
Right. So in terms of the ID tool, this was based on populations that extend pretty much as far north as Ottawa. So our confidence, we can say that's what we were covering. Of course, the further away you go, the potential for geographic variation to be more influential grows. But Northern Ontario, at the very least, some of the sort of general indications of which traits differ most strongly between the subspecies will still be useful. We've tried to put a good buffer on some of these traits, so it is as useful as possible over a large range. And in terms of looking towards Northern Ontario more broadly for the biocontrol work, this year we're looking, as I said, at expanding to at least 8 or more new sites, and some of those are further north in Ontario as well. So that's one of the locations that we're looking to expand into as well. So hopefully we'll be working in that area very shortly.
Okay, thanks again so much. That's all great to hear. There's a couple more questions for you in the Q&A if you wanted to just type an answer while we move on to our next presenter.
Will do. Thank you very much.
Thanks. Okay, so I would now like to introduce Elana Feldman from Utah State University, who will also be speaking to us about Phragmites. Welcome.
Is my screen shared properly?
Yeah, looks good.
Great. Thank you everyone for having me here today. So my name is Elana Feldman. I work under Dr. Karin Kettenring. I'm a master's student at the Utah State University Wetland Ecology and Restoration Laboratory. And I'll be talking about my thesis work on the effects of seed mix density and composition on invasion resistance to Phragmites. So quickly, I just wanted to give a plug for the lab. This is our lab website. We work directly with land managers to study the methods and mechanisms of wetland restoration. So all of our work is directly applicable to the work of land managers. So if you have any interest in working in wetlands, if you are currently working with either submerged aquatic vegetation or emerging vegetation, I highly encourage you to check out our website, check out our publications as to how you can better be managing your own wetlands with this research. So the agenda for today is I'll go over some research background. I will talk about some previous experiments that have been conducted in the lab that inspired my own research. I'll talk about some of my experiments that I've conducted by this point, and then I will give some final takeaways about how to apply all of this research to your own management of wetlands. So I study biotic resistance, which is the ability of a native community to resist invasion by an invasive plant, and I'm specifically interested in Phragmites. So we know that there are a lot of different factors that affect biotic resistance, but within my own research, I only look at density and diversity. So we already know that density is important because it allows us to overcome the high mortality that naturally occurs post-seeding. So even if a very low number of your seeds germinate, you'll still get high establishment. We also know that both density and diversity are important for filling niche space. So if we're already using all of the resources available in an area, then an invasive plant is not going to be able to come in and use those resources itself. What we don't know is if there are any interactions between these two characteristics that we can utilize when we're trying to create more invasion-resistant habitats. So when I'm talking about diversity in a community, I'm specifically talking about functional diversity, not species richness. And functional diversity is the diversity of characteristics that are present within a community, as opposed to just the number of species that are present. And I study this by using things called functional groups. So I use very broad groupings of plants with similar characteristics, and the 5 groups that I look at are listed here. And I also wanted to point out that I often use shorthand to refer to these groups. I'll call them either forbs or graminoids. So for anyone watching who is not a plant person, forbs just means plants with showy flowers. Graminoids are grass-like plants that don't have those showy flowers. So I'll quickly go into some previous experiments that were conducted by other members of the lab that inspired my own research. And these experiments answer the questions: does increased native seeding density reduce Phragmites growth? And which types of native plants reduce Phragmites growth? So I'll start with the work of Emily Tarsa, who graduated from our lab last year and now is a professor at the College of Coastal Georgia. This paper I highly recommend looking at if you are interested in managing Phragmites. She goes into a lot of different different factors in the ecosystem that can affect the ability of Phragmites to invade and persist. But one experiment she conducted that was important for my own research was looking at Phragmites planted at different densities along with native seeds planted at different densities and seeing which of these had a greater effect on the presence of Phragmites in a community. And her main takeaway was that Phragmites biomass is driven by the number of Phragmites seeds, not the number of native seeds. So this is important because although having a lot of natives in the community are going to be important for preventing Phragmites from entering the community, if you already have a lot of Phragmites in an area and have a dense seed bank, this is really what's going to be driving the presence of the Phragmites, and it's important that you continue to manage using chemicals and other means to reduce that seed bank instead of just relying on the native seeding itself to reduce the Phragmites. However, Emily also found that sowing more natives, so sowing at a higher native density, is going to lead to increased native biomass. So even if the natives aren't necessarily what's driving the Phragmites in your area, it's important to sow those at a higher density if you can, because that's going to help restore the native community there faster. So this research led to another series of experiments led by Ray Robinson, who graduated with her master's in the lab last year and now is working as our lab manager. So Emily's experiments only looked at graminoids. So Ray was curious about whether forbs would do a better job at reducing Phragmites in an area than the graminoids did. So Ray planted a series of different seed mixes that had different amounts of either forbs or graminoids in them to see if those differed in their ability to promote biotic resistance. So she had a 100% forb mix, a 100% graminoid mix, and then mixes in between that included both graminoids and forbs at different percentages. And she conducted this experiment in a greenhouse and in the field. And so she found that in the greenhouse, seed mixes with forbs successfully reduced Phragmites cover. So this was an important finding because in Utah, it's very common for land managers to only use graminoids, but this suggested that forbs actually have a lot of potential to reduce Phragmites in our area, and so that we should start incorporating them into seed mixes. However, when she conducted the same experiment in the field, she found that it was actually the graminoids that were reducing Phragmites cover, not the forbs. And so what this tells us is that both forbs and graminoids are important for reducing Phragmites, but they're going to be important under different conditions. So if you have an area that's very wet and has a lot of light, the forbs are going to help you out. Whereas if you have an area such as Utah, where it's often dry at certain points in the wetlands, that graminoids might do a better job than the forbs in those situations. And Ray also found that seeding at a higher density was always important for reducing Phragmites cover. However, the extent to which it continued to reduce cover beyond a certain point differed by site. So again, which site you are planting at is going to change how many seeds you need to plant. So this leads to the research that I'm currently conducting, which sort of combines all of this research, but looks at the question, how do native seed density and diversity interact to reduce Phragmites growth? So my first experiment was a greenhouse experiment that looked at the effects of native species identity and density on the growth of Phragmites. So in a greenhouse, I planted Phragmites with one of 18 different species that that represent each of the different functional groups I showed at the beginning of this talk. And I planted them at two different densities, either a low density or a high density, which I call here 1x and 5x. So 1x is just the density that land managers in Utah are already using, and 5x is 5 times that amount. And you can see from this picture right off that seeding at a higher density made a big difference in some of these tubs. Some species did a lot better than others. So here's a graph of my results. The x-axis is looking at all 18 of the species that I planted. The y-axis is change in Phragmites biomass. So anything that's below that red line means that the native plant that the Phragmites was planted with caused less Phragmites to grow. And you can see that some of those plants down by that arrow that I have had almost no Phragmites grow with them. So they had almost a 100% reduction in Phragmites biomass. So when I ran a generalized linear mixed model on this data, we found that species identity had a significant effect on Phragmites growth. So that just means that some species are better than others at reducing the growth, which you can see pretty clearly from this graph. I also found that high density significantly reduced Phragmites growth. So the higher a density you can seed at, that's likely going to help reduce the amount of Phragmites you have in an area. And finally, I didn't find any interaction between these two. So for right now, I recommend focusing on using the best species you can at the highest densities you can afford to seed at. But as you can see, probably some of these species, you could get away with planting at a lower density and still get some pretty good reduction of Phragmites. And similar to what Ray found, I found that the species that were almost entirely eliminating the Phragmites and preventing it from growing were the forbs. So once again, this is good evidence that forbs should be included in these seed mixes. And I'll get into later exactly what those species were that worked well. So here are some more graphs. These 4 graphs are looking at all the different relationships between Phragmites cover in biomass and native biomass in cover. And the main takeaway you should get is that all 4 of these graphs show a negative relationship, suggesting that when we have more native biomass in cover, we have less Phragmites biomass in cover. So the more natives you can get into an area, the less Phragmites we're going to have. And again, when we look at the species that were doing the most work at producing a lot of biomass and thus reducing Phragmites biomass, these were all native forbs. And these are the 4 species that at this point I would recommend using for restoration if it's native in your area. These were all very weedy species that produce pretty large leaves. So I am guessing that because they are just growing quickly and using up most of the light in the area, that is how they're preventing the Phragmites from growing. So my second experiment that I've conducted was a field experiment that was pretty similar, except instead of looking at the native species identity and density, I planted seed mixes that each represented a different functional group, and I planted those at different densities to see how these mixes would affect the growth of Phragmites. So the 5 mixes that I used are listed here. They're the same functional groups that I mentioned at the start of the talk. I planted at the 1x and 5x again, so the same 2 densities that I used for the greenhouse. And this had the added element of planting at 2 different sites. So one of my sites was at the Great Salt Lake, which, as the name implies, is a saline lake, and the other was at Utah Lake, which is a freshwater lake. So these are my results. So the x-axis is all the different treatments that I had. The y-axis is cover. Blue shows the native cover. Red shows the total invasive cover. So this was Phragmites and other invasives that came into my plots. And as you can see here, there are not a lot of clear differences between treatments. One of the main differences between these 2 sites is that they differ in their native cover. Utah Lake had much higher native cover, which suggests to me that freshwater lakes are going to be able to recover their native communities faster. Both of these were bare ground sites when I planted my seeds. So Utah Lake was just able to recover faster, and those seeds were able to establish faster than the Great Salt Lake was. You can also see that both of these sites had very low invasive cover, though unfortunately, I don't believe that this was due to my treatments. I believe that this was due to the weather. So this is an important reminder that the weather is not always going to cooperate with us, and it may take several years of seeding to be able to really see results in what you're doing. This is what my sites looked like at the beginning of the field season. We had a particularly unusually wet spring last year in Utah. And this is what they looked like at the end of the season. This is more typical of what we would expect in Utah. Plants senescing early, other things not growing because there's absolutely no water left in the wetland to be able to support those plants. So a good reminder for all plant scientists and land managers that the outdoors doesn't always do what we expect, and that's going to have an effect on our results. So now I'll give some takeaways for anyone interested in managing wetlands based on the research that I have right now. This is how I recommend you manage your wetland. First, higher seeding density reduces Phragmites. So seed costs a lot of money, so not everyone is going to be able to afford to plant at 5 times the amount they've already been planting, such as I did in my experiments. But I do recommend seeding at the highest density that you can afford to seed at, because it does seem to help reduce the Phragmites coming into an area. Second, forbs and graminoids both show promise for reducing Phragmites growth, just under different conditions. So I would definitely recommend including both of these in your seed mixes, maybe doing a small experiment if you can afford to do that on your own land to see which types of mixes are going to do best in your own area. There's no interaction between native seed density and diversity that I have found. So I would recommend focusing on just the best species for your area at the highest densities you can afford. And again, these results are going to be dependent on the field site. So the types of plants you should be planting, as well as the exact density you should be planting at, is going to be specific for each wetland. So again, if you can afford to take a year, do a small experiment on different sections of your wetlands, that's going to help you figure out what you should really be planting. So I'd like to thank all of my funders. Again, I recommend that you check out the lab if you're curious about our research, and I am happy to answer any questions.
Hey, great job. Thank you so much, Elana. It looks like we do have a question in the Q&A. Does the higher seeding density have to be specifically higher than the Phrag seed density? Also put, would it be useful to take a soil sample and look at the seed bank and Phrag seed density before buying seed?
So I can't answer that entirely. At this point, I would say that no, it doesn't have to be necessarily higher than the Phragmites like density in the seed bank. However, the higher the amount of Phragmites that you have in your seed bank, the more years this is going to take and the more difficult it's going to be. So that sort of gets back at what Emily's experiment was looking at. Really, the important thing that you can do is continue to manage the Phragmites, continue to deplete that seed bank at the same time that you're restoring the native plants, and you might not see a reduction in Phragmites immediately. But if you continue to encourage the native community to return and you try to prevent the Phragmites from reseeding itself, that's going to make a big difference eventually.
Okay, thank you. I think we maybe have time for just one more question. Is this applicable to, oh, sorry, this one was first. Sorry, I'm gonna read this one first. Have you done or plan to do any experiments on the impact of prescribed fire on these results? And do you think a burn would affect them?
So in Utah, we have a lot of air quality problems, so we're not really allowed to do many prescribed burns in this area. So that is something that I don't have a lot of information on, but I do know that that is something that people do to control Phragmites. I am not sure how it would affect the seed bank, but I imagine it could do a good job of removing a lot of the litter, which is one of the major factors that gets in the way of restoring native plants. Phragmites produces a lot of litter that completely covers the ground and prevents native seeds from being able to establish. So I would imagine that fire would help to remove that litter and then possibly help the native plants get back in, as long as the Phragmites seeds don't get there first.
Okay, and I think we can very quickly do just one more. Is this applicable to preventing Phragmites establishment only, or can it be used as a control method in an established site?
I would not recommend this as really a control method. This is really for areas that don't have Phragmites yet. So all of the areas that I work in are areas that used to have Phragmites, and it has been managed for about a decade at this point. So there's very little regrowth from rhizomes, and it's just regrowth from seeds that are blowing in from surrounding areas. So again, yeah, I would definitely recommend continuing to manage the Phragmites. That's going to be your main focus. And then once you have it removed, this is going to be an important factor that will prevent the Phragmites from returning once you've already removed it from the area.
Okay, great answer. Thank you again so much, Elana. So I'm going to introduce our next and final speaker of the forum. So welcome to Rob McGowan from the Ontario Federation of Anglers and Hunters. He's going to be speaking today about water soldier.
Thanks, Emily. I'm just going to get my screen up here. Here we go. Slideshow. Good afternoon, everybody. My name is Robert McGowan. I work with the Ontario Invasive Species Awareness Program. I'm the aquatic program specialist, and today I'm going to just go through a case study about the rapid response efforts for water soldier on an eastern Ontario lake, Red Horse Lake. Thanks for having me here. So let's just get started. So just a bit of an overview. We're just going to go through what water soldier is. Why it's a problem. We're going to get right into kind of the rapid response efforts in 2020 when we got the information that water soldier is present in Red Horse Lake. And we're going to just continue from there and see how it's played out since then. So what is water soldier? So water soldier is an invasive perennial aquatic plant that's native to Europe and Northwest Asia. The first known wild population in Ontario was discovered in September 2008 in the Trent Severn Waterway. Water soldier was sold regularly as an ornamental plant in water gardens, which is a likely source of its introduction to the Trent River. And so why is water soldier a problem? Well, it forms dense mats of floating and submerged vegetation. It crowds out native vegetation, which results in reduced biodiversity. The dense floating mats of water soldier hinder recreational activities such as boating, fishing, swimming, hunting, etc. And the sharp serrated leaf edges can cut swimmers and individuals who handle the plant. So you can see this picture on the right, those serrations on the leaf margin, they can do some damage. So we want to, if you're handling the plant, you want to be gentle with it. So as we said, water soldier was first reported in the Trent Severn Waterway in September 2008. It spread down much further than 50 kilometers now, and it's been found in the Bay of Quinte. So you can see the Trent Severn Waterway runs from Georgia Bay or Lake Huron down to the Bay of Quinte, Lake Ontario, which is in Trenton, and we have unfortunately found it in the Bay of Quinte now. So another second wild occurrence was in the Black River in 2015. Rapid response efforts by the Aurora District MNRF and Peterborough District MNRF and OFAH and the Invasive Species Center have hopefully eradicated it. We haven't been able to find a physical plant since 2016. And Red Horse Lake in August 2020 was the third wild occurrence in Ontario. It was it's north of Gananoque or north of Kingston. So here this is a pretty popular timeline series that a landowner has kind of documented for us. And we're going to just give you this bit of a timeline and show you why water soldier is such a problem, why we don't want to take our time to manage it or just sit on our hands for a bit. So you can see here, this is summer 2013. You can see the water soldiers present along the shoreline and a bit out into the bay here. So here we have 2014. So it's a lot denser now. It's starting to fill in all the littoral zone there, and it's starting to spread out. And you can see that the landowner has to change her perspective now. So now it's gone beyond her dock. So all of this area has been filled in. And here in August 2015, you can see that it's pretty much taken over the bay. So this plant establishes very quickly. It reproduces very quickly through offsets and turions. And this is why once we get a report, we want to act as fast as we can. So the Red Horse Lake report was called into the Invasive Species Awareness Hotline on August 27th. A concerned citizen that has interest or lives on the lake came across the plant, didn't recognize it, did a little homework, and I guess he Googled it and found out that it was water soldier, and he sent us some pictures, and we confirmed it was water soldier, and we started communicating with MNRF to see what we can do about it. So Red Horse Lake is located in Eastern Ontario, halfway between Toronto and Montreal. It's 24 kilometers north of the 401. Red Horse Lake is one of a 28-kilometer long chain of lakes that comprises the upper Gananoque River system. And if you're still kind of not really knowing where you are, it's right beside Charleston Lake Provincial Park. So the first steps to a rapid response. So we have a protocol that when we get a call about priority species, which water soldier is listed on the Invasive Species Act and listed as a prohibited species, we have to document, verify, and then communicate with MNRF to make them aware that we have a report and it's confirmed. So we did that. We had a video conference because this was during COVID. We really didn't know what we were going to do, if we're going to be able to get out on the land and actually do something about it. But it was decided that we were going to. So a few protocols were written up, and we headed to Red Horse Lake. So when we got to Red Horse Lake, we conducted a site visit. The site visit informed us of the size and density and number of populations of water soldier that we would have to hopefully eradicate. Although the population was larger than we thought, it was isolated to one population. So that was kind of good. And you see the population here. It's really deceiving. It really is much larger than what it looks like, because the water the depth drops off very rapidly. So some of these plants that are in deep water weren't emergent. They stayed submergent. So when we were there, we delineated the population, and we also took some eDNA samples. I don't know if you saw Colin Cassin's presentation yesterday. eDNA is a early detection tool. Even though we knew water soldier was there because we physically went there, we took some samples upstream and downstream of the populations and to see if we got any hits there. The biggest hit we got was obviously at the site of the population. So the rapid response. So on Tuesday, oh, sorry, wrong page. So after we did our site visit, we went back and further discussed with MNRF and Invasive Species Center what our steps forward are going to be. And through our lessons learned dealing with water soldier and Trent Summer Waterway and on the Black River, we decided to move forward with a herbicide treatment. So in October of 2020, we hired a herbicide applicator to go treat this population with diquat dibromide, and which is the same product that we treat the water soldier with on the Trent Severn Waterway. So the herbicide treatment happened. Graham Agri Services conducted the treatment. And skip ahead to 2021, we revisited Red Horse Lake to do more surveillance and see how the treatment turned out, to see what the efficacy of the treatment was. And you see on the left here was the initial pictures taken in 2020. And on the right is the 2021 post-herbicide pictures. It may not look like it. There was less water soldier. And this is pretty typical of what we're used to after treating water soldier for 1 year. You see a bit of damage to the leaves of the plants, and there's not as the density isn't there like it is. It's usually after the 2nd year we start to see some real changes in the population. So 2021, we really started hitting our outreach education with the lake associations, the Charleston Lake Provincial Park, local MNRF districts, to let them know that water soldier was present. It was being controlled, but it just to get the word out there to see if anybody else has noticed it anywhere else in the area, because you can obviously see from those timeline pictures how fast the plant can establish and grow. So, water soldier 2022, we revisited water soldier Red Horse Lake in 2022, and you can see that the majority of the water soldier is gone after the 2nd treatment. These pictures are a bit deceiving. There's still water soldier there. But the decision was made to not just do surveillance any longer. We implemented the water soldier monitoring protocol overlaid the grid onto Red Horse Lake, and we systematically sampled the lake. So you can see here, basically, we had the lake, overlaid the 50 by 50 meter grid. Each point intercept has a unique identifier, and we collect certain information at each point intercept. So we collect the water depth, the water soldier value from 0 to 3, 0 being no water soldier, 3 being dense populations. And we also collect some community information. So the top 3 species of aquatic plants that are present along water soldier. And this is useful because now year after year, we can compare how successful our treatments are and how the plant community is changing, or if water soldier is even present anymore, which is our goal, to eradicate water soldier. So you can see here, we did have some water soldier found. The orange dot indicates scattered plants, and the red dot indicates a dense colony. This is within the same area that the original population was found, so it wasn't surprising to us. But as you can see, in the upstream of the population and downstream from population, we didn't find any other plants, which was good news. So we delineated the area. As you can see here, this is about 0.5 hectare area of area to be sprayed with diquat dibromide again in 2022. And in October, it was sprayed. So moving forward, in 2023, we're going to be going to Red Horse Lake to monitor again with the monitoring protocol. We'll be conducting surveillance upstream and downstream of the populations, and we're going to continue with our outreach and education. And one last note is that none of this is possible without the collaboration of the MNRF, Invasive Species Center, Invasive Species Awareness Program, and the cooperation from landowners. It really like to thank everybody because it wasn't an easy task just putting all this together during the height of the, well, not the height, but during the pandemic. And we're hoping that moving forward, we'll see a success in eradicating water soldier from Red Horse Lake. So thank you very much.
That was great. Thank you, Rob. We've got some time for questions. We're right on schedule. So first question I have is, how did water soldier get into Red Horse Lake?
Well, we don't know, but again, it was very popular in the water garden nursery industry. And even though it was listed as a prohibited species in 2015, these plants, the way they grow, there's so much of it. And there must have been so much just sharing of the plants, because you'd have this in a pond, and then the next year the pond would only be water soldier. So I know some of these water garden agencies and stuff, they were just giving this plant away when you made a purchase of a fish. And when the Invasive Species Act did come in, we had some of these nurseries calling us saying, well, we got 10,000 plants here. What do you want us to do with them? So it was pretty popular in the water garden trade. And so I still get calls because Invasive Species Awareness Program does manage private ponds across Ontario. And we've managed 16 to 17 ponds since 2015. So every year we find another pond with water soldiers. So they're out there.
Okay, thank you. Another question we have. We've been hearing about rapid response for water soldier for 10 years in that area. So why is it still there?
So this is not the Trent Severn Waterway. The Trent Severn Waterway runs from Georgian Bay down to Bay of Quinte, which is Trenton. This water soldier in Red Horse Lake is about 250 kilometers away from the Trent Severn Waterway. So it's in a new area. It's we just can't have it establishing. Basically, the Gananoque River system runs down into Kingston, which is the mouth of St. Lawrence River. So we don't want this plant in another major waterway. So the rapid response is to this population specifically.
Okay. Next question. Is anybody charging those water garden suppliers? I guess since the act came into place.
I have not heard of anybody being charged. I have worked with enforcement to help identify and remove water soldier from some ornamental ponds, but I can't speak to any charges being laid.
Okay. And it looks like we have one more question here. Has it been eradicated in the Trent system yet?
It has not. We are working on it still. It's a very different plant to get rid of. And it being in a flowing system like the Trent Severn Waterway is, we play catch up sometimes. So we're seeing great success in the upstream populations, but with the lock system, there's on Ontario Hydro waterways, it's very difficult to control how the plant moves through the system. So it did get into the Bay of Quinte, and we're monitoring that this year. And Parks Canada right now is developing the next 5-year plan from 2023 to 2027. So we are still going to be managing the water soldier in the Trent Severn Waterway.
Okay. It looks like another question has just popped up, which we still have time for. I guess just asking about mechanical removal versus chemical removal.
Yeah. Well, you see the picture up on the screen. We've done lots of hand pulling. We've had a pilot control project completed with the mechanical harvester. What we found is that the mechanical harvester is expensive. We don't own one, so we have to hire a company to come do it. We didn't see that great of results, and that may just be because of the Trent Severn Waterway is a very shallow waterway. Where water soldier grows is usually in back bays. There's lots of stumps and logs, and it's just difficult to navigate. And you spread the plant if you're not careful. So you have a large machine coming in, scooping up these plants, and you're stirring up all the sediment. Plants aren't going to get, they're going to get dislodged and they're going to get lost. So you have muddy waters now, and you have a plant that floats, and it can be buoyant in the water column, and it'll just float away. So we find with herbicide that we don't disturb it, and we hit it hard, and we see great success. And if we do a cost-benefit analysis, the mechanical harvester can be more expensive than the herbicide treatment.
Okay. Well, thank you very much. It looks like that's all of our questions. So thanks again for joining us today.
Thank you very much.
Okay, so we are coming to the end of our forum. I do just want to thank everyone for joining that session and for joining us in the forum. Before I move on to our closing remarks, just want to touch on the Green Shovels Collaborative. This is a collective of like-minded conservation organizations and a collection of projects designed to achieve job creation, economic recovery, and environmental progress while addressing invasive species issues. So you can check that out at greenshovels.ca. Also want to ask everyone to please provide your feedback. It's really valuable to us, so take the time to fill out the short survey on this event. It'll be sent out to you afterwards. That way we can know what you liked about the event and how we can improve moving forward. So you will get that in your email inboxes, and you can always see more of our upcoming events by visiting our website, invasivespeciescenter.ca. Thank you.
Awesome. Thank you so much, Em, for taking us through the afternoon today and our final afternoon of the forum. As she mentioned, please fill out our post-event survey. We do read every comment and we make adjustments every year to make the experience better for you, our attendees. And we had some new social and networking initiatives this year, so your feedback is going to be especially important. Before I pass it over to Sarah to give our closing remarks, I'd like to thank everyone who made this forum possible, including our moderators and everyone on the Invasive Species Forum team. I'd like to thank the Ontario Ministry of Natural Resources and Forestry for their continued support of not only this event, but much of our work at the ISC. And I'd also like to recognize Lalaman Plant Care, a bronze sponsor of our event this week. Lalaman does have an ongoing webinar series, and next week's webinar will be focused on the use of triazin for hemlock woolly adelgid management, which I think will be relevant to much of our audience here this week. So try to check that out if you're available. And finally, a big thanks to each and every one of you for joining us over the past 3 days. We really appreciate your support and hope you learned a lot this week and can take some things into your own work. With that, I'm going to pass it over to Sarah, our Executive Director at the Invasive Species Center, to give our closing remarks for the event.
Thanks very much, Mackenzie, and thanks to all of you in the audience. We're reaching the end of the 3-day Invasive Species Center hosted annual forum. It's our 4th forum. My name is Sarah Rang. I'm delighted to be the Executive Director of the Invasive Species Center. Just want to echo Mackenzie's comments to thank our sponsors who've made today possible and enabled the free registration that I think so benefits all of us. So that's the Ministry of Natural Resources and Forestry and Lallemand as well. We've had over 60 speakers. I'm hoping that they entertained you and inspired you a little bit. We've also been delighted to have record attendance over the past 3 days with over 900 people registered. We've traveled the country together virtually. We've learned about forest and aquatic and plant invasives. We've been exploring our theme for this year's conference, which is Invasive Species Action in a Changing Climate. And so we've learned together some of the main interactions between invasive species and our climate, and often resulting in increased spread and pressure from new invasive species. We were honored to host some of the Indigenous communities and hear about new projects on invasives and the importance, the reminder really of the importance of considering two-eyed seeing in all of our approaches. We've heard interesting new work from municipalities on mapping and monitoring, and also how to set priorities for the management of invasive species and how to integrate these into municipal programs. Thanks to all the scientists and the community groups who've joined with us and shared their sense of what's going on in the lab and on the ground, and how to actually integrate invasive species management into many land management activities. We've heard about new innovations and tools, and some of these from drones and mappings, management, and the all-important communication of invasive species. In the awards section, I hope you joined with us to learn about the many individuals and organizations who are inspiring us with their ways to make a difference. And a big thank you to our keynote speakers, Gail Kranzberg, who challenged us to collaborate on science for society, Jeremy Alliston and Josephine Korlek from Natural Resources Canada, who shared ideas on forest biodiversity. And finally, today we heard from Bethany Bradley, who helped us understand and tease out some of the interactions between climate change and invasives, and gave us some great new ideas in terms of the power of people to connect ourselves and horizon scanning. So powerful and inspiring ideas for all of us. As noted, we'll be seeking your feedback. Pleased to hear your suggestions for next year's forum. And if you missed any of the sessions, don't worry. Please, you can see them again in terms of the tuning into the Invasive Species Center YouTube channel, which will be posted shortly. And I just wanted to highlight some of the ways that we have to continue the conversation together and continue in terms of exchanging great ideas. We are delighted to host with many different partners the Invasive Species Awareness Week, which is February 20th to the 26th. It's your chance to post your ideas about invasive species management, trying to tell the story of invasive species to a broad audience. So if you need any free materials, just check out the ISC Invasive Species Center website and many of our partners' websites as well. In the spring, we'll be opening our Invasive Species Center microgrant program. So this is a chance to receive funding for local community action on invasive species management. And final event is our International Conference of Aquatic Invasive Species, which is May 12th to the 16th of 2024. So a little advance notice there. It's a great opportunity to join together with experts from all around the globe and learn more about aquatic invasives. So as we close the forum, a big thank you and shout out to our speakers who shared the knowledge. A big thank you to our hardworking Invasive Species Center team who put the event together, and both in front of the camera and behind. So that's Deb Sparks, Mackenzie, Lauren Rogers, Lauren Bell, Colin Casson, Madison, Darissa, Nicole, Leanna, Rebecca, Tara, Emily, and many more. And a big thank you to you for sharing your time with us. We're always looking for new partners and projects, and so please don't hesitate to reach out to any of us if you'd like to collaborate with the Invasive Species Center. And we truly believe that together we can make big strides forward through the power of collaboration and look forward to the next decade together. So I hope the forum's provided you with some new insights, perhaps a new friend or two, or a new partner to collaborate with, and most importantly, to be inspired to continue your important work on invasive species management. So I'd like to now declare the 4th Annual Invasive Species Forum closed, and look forward to participating with you in the many other ways that we can throughout the year. Big thank you to all of you. Let us know if you have any questions. Thanks so much.